Humans have two skins, the one that you can see and the one you don't. The skin that you don't see is formed by an army of bacteria, viruses, and yeasts that colonize your body shortly after your birth, and they live with you, or rather on you, until you die. These cells protect you from pathogens that vie to infect your body, but under certain circumstances, they become pathogens themselves and cause diseases like cavities, abscesses, shingles, and cellulitis. This video will discuss the role of these microorganisms and others in your and my lives. By the end of this video, you should be able to give a brief description of the normal flora and its role in human health. You should be able to distinguish between the words infection and disease, and you should be able to distinguish between emerging infectious diseases and re-emerging infectious diseases. It's estimated that there's somewhere between 50 and 100 trillion cells in the human body. So that's something like a 1 with 14 zeros behind it. It's a huge number. But believe it or not, there's actually 10 to 20 times more bacterial cells than human cells in the human body. A famous scientist once said that if an alien were to come to this planet and look at a human being, they would assume that we're bacterial, based upon the number of bacterial cells that are on our body versus human cells. The question is, what are these microbes doing? Your normal microbiota, also called the normal flora, is made up of microbes in your body that set up permanent residence. That is, they colonize you under normal conditions. And microbes start doing this as soon as you're born. And then with every interaction that you have with your environment or with people in your environment, you pick up more microbes and they continue to live with you. Now we know that the populations of some microbes changes over time, but from the day you're born till the day you die, you'll have these microbes with you. Some of the microbes are forming what's called a biofilm. A biofilm is basically a large community of microorganisms. As you can see in this picture, there are cells that are coccus-shaped, there are thick bacilli, long bacilli, short bacilli, thin bacilli, all sorts of organisms, and they're embedded within a special kind of substance called the EPS, or extrapolymeric substance. It helps hold the cells together and affix them to a surface. We grow a biofilm every morning on our teeth. It's that transparent, slimy layer on your teeth. So if you take your tongue and you run it across your teeth right now, the surface of your teeth probably feels kind of smooth and slippery. Well, that's the biofilm. When we fail to remove that biofilm for a number of days, we may develop this. This is called dental plaque, and it's an extra thick layer of biofilm. That can be removed with brushing and a visit to a dental hygienist. Most of the studies that are looking into the effect of normal flora on health are done on germ-free animals. So these are animals who are born with no normal flora, and then scientists add different groups of bacteria to those animals and look to see what happens. So these studies on germ-free animals have suggested that bacterial colonization of the gut is important to the maturation of both the enteric nervous system and the central nervous system. We also seem to be finding that the absence of microbial colonization is associated with altered expression and recycling of neurotransmitters in both nervous systems. Studies on germ-free animals have also demonstrated that the normal microbiota influences stress activity and anxiety-like behavior for these animals. And we know that the gut microbiota has a role in obesity. So typically people who are obese or overweight have a lower fecal bacterial diversity than people who are normal weight. We know that some of the bacteria that are present in overweight and obese individuals, they are associated with more inflammation and um, increased extraction of nutrients from food. So that may contribute to the obesity. The microbes in the gut play a lot of normal, important functions. They help produce energy that we ordinarily would not be able to access because we don't have the enzymes to break down those nutrients in food, things like soluble fiber. We know that these organisms can make vitamins for us like biotin, folate, and vitamin K. They help metabolize some of the chemicals that are produced when we cook food. They coat our intestines and provide a barrier that pathogens have to essentially get around or get through in order to make us sick. And they also help in developing our immune system, our gut immune system. There's also some speculation that the normal flora may play a role in aging. So it's well known that the composition of the normal flora in young adults is very different and more diverse than that of older adults. Experiments in animals suggest that the shift in normal flora happens before we actually start aging. So this would mean that some of the microbes that grow while we're just before we start aging, they're associated with increased inflammation and changes in metabolism. So that may, play a role in how we age, how fast that happens. Now, in addition to being part of the normal flora, occasionally some of the microbes that are part of your normal flora may become pathogens. We also come in contact with pathogens just moving around in our environment. A pathogen is a microorganism that can cause disease. Diseases that are caused by pathogens are called infectious diseases. So here a pathogen invades a host and then it begins to grow inside of that host. And something about the growth of that microorganism leads to a change from the state of good health, which is what a disease is. Now, in order for all of this to happen, the pathogen has to get past our body's resistance factors. Resistance refers to the ability of our bodies to ward off disease. And factors that contribute to our resistance include things like the skin, which serves as a barrier against pathogens. You have proteins called defensins and lysozyme that basically attack the bacterial cell wall and other parts of bacteria to kill them and inhibit their growth. Lysozyme is in your tears. It's also in your sweat. You also probably are aware of the acid mantle, that the skin has a pH of around 5.5 or so. This is a pH that is hostile to the growth of bacteria, so that's going to, again, inhibit the growth of bacteria. You also have proteins like fibronectin, which helps form blood clots in a roundabout way. And those blood clots isolate bacteria so that they can't spread through the body and cause disease. So there's a number of factors like this that protect you from disease. When you get an infectious disease, it's because a pathogen has overcome all of those resistance factors. So let me give you an example of how this happens. This is a case study from 2017 about an event that really happened. There was a 31-year-old Latino man. He had a history of chronic liver disease and he decided to get a tattoo. Now, as I mentioned already, one of the body's best defenses against infection is the barrier protection offered by the skin and mucous membranes. So when you get a tattoo, you effectively punch hundreds of tiny holes into that protection, and it can make someone susceptible to an infectious disease. So the man gets a tattoo. You can see it here in this picture, lovely tattoo of a cross with praying hands and script around it. About 5 days later, he decides to go swimming in the Gulf of Mexico. Now, the Gulf of Mexico is part of the ocean. He's basically taking a dip in a bacterial soup. This is the same reason why a tattoo artist, for example, tells you don't take a bath for a certain amount of time after you get a tattoo, because soaking in a bath, you're basically soaking in a bacterial soup. You're opening yourself up to infection. So anyways, about 3 days after this man goes swimming, he shows up at a hospital with severe pain in his legs and feet. After an examination, he gets admitted. Within a few hours, the skin on his legs starts to darken and bruising appears. This is followed by discoloration and the appearance of bullae. So bullae are mounds of fluid that happen under the skin. The doctors determine that he's in the early stages of septic shock, and they start giving him antibiotics because they suspect he's infected with a bacterium that's common in the Gulf of Mexico, the water, the ocean water. It's called Vibrio vulnificus. So they give him these antibiotics based upon an educated guess, based upon what symptoms he has, as well as knowledge of what types of bacteria he may have been exposed to. Septic shock is kind of the third stage of disease that happens after sepsis. Sepsis is a life-threatening complication of infection when chemicals released by the body to fight that infection actually trigger a severe inflammation response throughout the body. So you could think of sepsis as being a severe inflammation response, and it's step 1. Step 2 occurs if the infection is not under control after a bit of time, and it's called severe sepsis. Here, the infection reaches the bloodstream and it causes inflammation, but the inflammation is severe enough to affect the function of organs like the kidneys, the heart, and the brain. Current thought is that the inflammation maybe causes tiny blood clots in those organs, and so they can't function properly. If the infection is not controlled after severe sepsis, then that's when septic shock can develop. So here you get a significant drop in blood pressure, and that can lead to respiratory or heart failure, stroke, kidney failure, or the failure of other organs. If septic shock is not brought under control, then death can result. So the man gets put under sedation. He's intubated, put on a respirator, and his tests come back. They find out that he does indeed have an infection with Vibrio vulnificus. It's a common coastal bacterium. You find it in ocean water. It causes about 8,000 illnesses every year and about 100 deaths, mostly among people who are already sick with something like diabetes, HIV, cancer, thalassemia. Remember, this man had chronic liver problems, so he was susceptible, and then his chronic liver disease was like an added complication. Around day 27 after being admitted to the hospital, the doctors are thinking that he's doing a little bit better, so they take him off of his ventilator, and not too long after that, his condition starts to worsen. About 2 months after he had been admitted, his condition is worsened enough that he dies. And he dies of septic shock. So in the picture down here, you can see how the discoloration is happening. You can see the bullae, the little bubbles here, and you can see what's happened here at his tattoo. So the progression from sepsis to septic shock was only 12 hours. And this was after the doctors had already given him antibiotics. So he was in it was a really terrible situation for him. So scientists and public health officials will sometimes classify infectious diseases based upon whether or not they're new or they've been around before. Emerging infectious diseases are diseases that have not occurred in humans before, like the SARS-CoV-2, or they've occurred previously but only affected maybe a small number of people in some isolated areas, like Ebola, for example. Remember, it showed up in different parts of Africa, but it mainly affected small, isolated villages. Or these are diseases that have occurred throughout human history, but we didn't know what was causing them. We've only recently uh, figured out that they're distinct diseases and identified the microbe that's responsible. So Lyme disease would be an example of that. So emerging infectious diseases can be caused by a number of different factors. One could be, for example, maybe people are traveling to new locations. Someone decides to go visit a remote um, a remote village in Africa or Indonesia, and they become exposed to a new microorganism. Then they carry that microorganism back to the city where they live and they pass it on to other people. That can cause more and more people to become exposed. It could be that people are traveling into new areas that they've never been in before, like maybe they're chopping down rainforests, and there are microbes in the rainforest that you don't usually find in populated areas. People become exposed to new animals that way, or new plants. Maybe it's through drinking water. They pick up a new microorganism and then they take it back to their home and pass it to people in their household, and then those people pass it to other people in the neighborhood, and then people in the neighborhood travel to other cities, pass the microorganism, and it's kind of like what we saw with SARS, really. And then human behavior and society, how we travel and and spend time with so many different people can also facilitate the transmission of new diseases. This is a slide that basically gives you some examples of what were considered emerging infectious diseases back in about 1995. So it's kind of an old slide, but um, some of the diseases that are on this slide would still be considered emerging. Re-emerging infectious diseases were major health problems at one time, then, say, through vaccination programs, the incidence and prevalence of those diseases precipitated. You went from having lots of cases to having only a handful of cases. But now, those diseases are becoming a problem again for a number of people. There are a number of factors that can lead to re-emerging infectious disease. The first is development of resistance to antimicrobials. An example of this would be malaria. Malaria is a disease usually associated with tropical areas like parts of Africa and parts of Asia, South America. Basically, malaria is a protozoan that infects red blood cells. It can cause fever, chills, sweats, headache, and vomiting in the uncomplicated form. In the complicated form, you get organ failure, anemia, cerebral malaria, which can cause altered mental state, seizures, coma. Um, you get blood abnormalities, ARDS, that acute respiratory distress syndrome that we see also with COVID-19. That can be caused by malaria. You can have shock, all sorts of really nasty effects. And so there are a few treatments that we use for malaria. Chloroquine is one. There's another group of drugs that are also used, but the malaria parasites are actually developing resistance to both drugs. So that can cause a reemergence because those drugs are not as effective anymore. Sometimes the organisms change themselves. I think about influenza viruses. They have what's called a segmented genome. So that means they have multiple strands of RNA that make up their genetic core. Well, when one organism is infected by two different types of influenza viruses, those viruses can switch segments of segments of RNA and make new viruses. So that can cause reemergence. Breakdown in sanitation or animal control If you think back to around 2009, there was a major earthquake that happened in Haiti. For about 100 years before that earthquake, Haitians had not seen cholera epidemics. Because the earthquake destroyed the sanitation system and you suddenly had lots of broken latrines, that meant that groundwater and the water that people was drinking became contaminated with fecal matter. And unfortunately, cholera outbreaks began in that country again. And then there's inadequate vaccination. Here I have to think about the United States. Prior to about the 1970s, you had lots of childhood illnesses like measles, whooping cough, uh, polio, I think, was on its way out by then. But you had a lot of childhood illnesses, and we had these great vaccination programs that basically took the cases of these childhood illnesses from the thousands down to the hundreds and lower. Nowadays, people don't want to vaccinate their children because sometimes they're afraid of what's in the vaccine or they don't understand how vaccines work. And so we're starting to see the reemergence of childhood diseases like measles and whooping cough again. So this is a slide that shows some examples of re-emerging infectious diseases. So that brings us to the end of this chapter. I hope after watching this video you can give me a brief description of the normal flora and some ways that it is important to human health. Remember, we said the normal flora is made up of those microorganisms that live in and on your body. You became exposed to them shortly after your birth, and basically they're living with you now. They are colonizing your body. Mostly they're serving a protective function, but remember, under certain circumstances, they can become pathogens. We also talked about how microbes that are in the gut produce chemicals that can change our brain activity, basically. We briefly talked about how organisms in our gut can change how we absorb nutrients from the food that we eat and can make us overweight or make us underweight or healthy weight. We talked a little bit about infection and disease. Remember, infection is when a microorganism is able to overcome your resistance to infection, and then that microbe starts to grow in the body. An infection can progress to disease when there is a change from the state of good health. So, an infectious disease is when a microbe gets into the body and it grows, and then that leads to a change from the state of good health. We also talked about emerging infectious diseases and re-emerging infectious diseases. Emerging infectious diseases are ones that have recently surfaced. They are brand new. They are things like the SARS-CoV and H1N1. That's a type of influenza. Re-emerging infectious diseases are diseases that are reappearing in new forms. So, for example, maybe they're antibiotic resistant or they're showing up in new places. And two examples of re-emerging infectious diseases would include the drug-resistant malaria and drug-resistant tuberculosis. There are others. It's just those are the two that I'm thinking of as examples right now. In the next video, we're going to take time to drill down and study the prokaryotes in more detail. I'll spend a lot of time talking about how prokaryotes are different from the eukaryotes that we're already familiar with. I look forward to having that chat with you later. If you have any questions about this chapter or any other chapter, feel free to email me or text me and we can have another discussion. I'll see you at the next video.